When you build a building, you have a set of blueprints, and an architect and engineers sit down and design a communication system, plumbing, air conditioning, all of that. In the brain, we're actually trying to reverse engineer that in a way. We actually don't have the blueprint.
Possibly the most complex entity in the entire universe. There's microarchitecture, nanoarchitecture, macroarchitecture, and then there's the function of the brain, which we observe in everything we do when we interact.
As humans, we have this intellectual curiosity about what makes us different. It's all related to our brain circuits. Getting the tools to allow us to see circuit activity in action and then apply it to problems that no one's been able to solve before would be a tremendous advance.
We all have a brain, and we want to understand that brain better. Get the scientists from different communities, different cultures, different emphases together. They'll start forming their collaborations organically. To do that type of sharing, you don't need a big bureaucracy. You don't need a lot of complexity. You just need to talk to each other.
No one person or even small number of people have the expertise in terms of anatomy, physiology, computation, theory, mathematics. We can all act synergistically to come to a better model of what's going on.
Every one of us have been touched either directly or indirectly by mental conditions. And the fact that we don't have answers for a lot of these things, we don't know how to cure depression, we don't know how to treat Alzheimer's, we don't know how to deal with post-traumatic stress disorder. When you have diversity in a group, you would take in the advice, experience, expertise from others, and that, I think, at the end will end up enriching whatever you're trying to build.
Brain Initiative combine the science and engineering together to develop the tool which can impact the neuroscience community, interrogating the signal connectivity, how these single neuron will be connecting to the next neuron, how memory is being really formed and retrieved, deeply understand the last frontier of our mystery of the brain.
For researchers, oftentimes there's questions of funding, there's questions of relevance, and foundations can say, we're committed to this work because we're doing this work. We're putting our mouths where our money is and our money where our mouth is.
If we as a government don't fund basic science, who's going to do it? Better outcomes for patients and for all of us, whether you are diagnosed with a disease or not. It's going to save us a lot in the long run as far as healthcare costs.
Let's get all the stakeholders, industry, government, academics, lawyers, doctors, people from all parts of life, and let's come up with a structure to help people in the greatest need.
What would happen if we put this, and that together? If it actually worked, it could really advance things. And in terms of the research that we do in neuroscience, I think the Brain Initiatives enabled that kind of high risk, high reward.
We should just get people together to pursue this goal that is much greater than any one of us individual professors or scientists. What could be more interesting than understanding what the ingredients to consciousness are? That's our vision.
Most detailed human brain map ever contains 3,300 cell types. Scientists just unveiled the largest and most detailed atlas of the human brain ever created. It details the arrangement and inner workings of 3,300 types of brain cells, only a fraction of which were previously known to science. The research was released Thursday, October 12th, in the form of 21 new papers published across 3 journals: Science, Science Advances, and Science Translational Medicine. It's not just an atlas. Ed Line, a neuroscientist at the Allen Institute for Brain Science and a lead author of 5 of the papers, told MIT Technology Review. It's really opening up a whole new field where you can now look with extremely high cellular resolution in brains of species where this typically hasn't been possible in the past. These cells include neurons, the brain cells that communicate through chemical and electrical messages, and a roughly equal number of non-neuronal cells. These non-neuronal cells include glia, a class of brain cells that provide structural support, nutrients, and insulation to neurons while also regulating how they send signals. The adult human brain contains an estimated 86 billion neurons, give or take about 8 billion, and another 84 billion or so of these non-neuronal cells. The BICN Human Brain Atlas used cutting-edge techniques that had previously mostly been used in animals. Mattia Moroso, a senior editor for the journal Science, wrote in the special issue published Thursday. Scientists used transcriptomics, which involves cataloging all the RNA in individual cells, RNA is a genetic molecule that contains instructions to build proteins and does other important jobs. They also used epigenomics, which involves examining chemical tags that sit on top of DNA and control how genes can be used. Single studies included in the BICN included data from hundreds of thousands to millions of brain cells. His allowed for a direct comparison between human and non-human primate brains and revealed that the myriad cell types found in our brains are also found in chimps and gorillas, the New York Times reported. But although we share cell types, the gene activity of those cells appears to be markedly different in humans and apes, and this changes how those cells work together. It's really the connections, how these cells are talking to each other, that makes us different from the chimpanzees. Trygve Bakken, a neuroscientist at the Allen Institute who worked on the primate studies, told The Times, although unprecedented in its detail, the Human Brain Atlas is a first draft. In the future, scientists want to decode the function of the newly discovered cells in the brain, many of which lie deep in the brain, in structures like the brainstem, Nature reported. They also want to understand how the gene activity of different cells contributes to the development of neurological diseases. In a statement that ran alongside the 21 newly published papers, Science said, if we want to understand what makes us human and the mechanisms responsible for the development of neurological disorders, we first need to have a deep knowledge of the human brain at the cellular level, which is exactly what this collection of papers from the BICN is about.